Well, thank you everyone for taking the time to come listen to this talk. I'm really excited to give it. So my name is Erica Griggs. I'm a PhD candidate at UVM in the biology department, and I work with a lab group called the Wildlife Pathogens Lab, which is run by Dr. Ellen Martinson. You should check out our website. We do a lot of really cool things in the lab involving parasite and pathogen research in native wildlife of Vermont. So we have a lot of small animal projects going on, looking at tick-borne diseases. I have a peregrine falcon project and another raptor project, so stay tuned for that. But tonight, I'm going to be talking about tracking avian malaria parasites and common loons right here in New England. But before I begin, I just want to do some acknowledgements. So my funders, the Nuttall Ornithological Club, has been really generous with their funding for this project. I'm also funded by the Vermont Space Grant for this work and the NSF GRFP. And I have some really amazing loon collaborators in the Biodiversity Research Institute, Adirondack Center for Loon Conservation, and the Loon Preservation Committee, who without them, their time and their support and their samples, this project wouldn't have happened. So before I get into what I'm studying, I just want to talk about really quickly that global change. So this figure that I have here, or this picture, is just a map of how the air surface temperature has changed over the past 50 years. And so you can see that the climate is warming, and what that's doing, besides creating more intense weather events, it's also changing the dynamics of vector-borne diseases, specifically how the vectors themselves are behaving. So vectors, I'm talking about mosquitoes, biting midges, and other insects that carry diseases, are actually increasing with intensity when the climate changes and with the warming. And also they've been moving their direction into northern latitudes. And so what we're seeing is a public health crisis in people, but also in wildlife, where hosts that normally haven't been interacting with these parasites are now encountering them. And then we're having this feedback loop of the climate keeps changing, vectors change their behavior, which impacts hosts who potentially haven't encountered them before, and overall feeds into a decline in biodiversity for some species. And the perfect species to study that in is our common loons. So loons have been sentinels of aquatic ecosystem health for the past couple decades. And this really cool quote from the Loon Preservation Committee, which is a healthy and stable populations of loons with good reproductive success indicates a healthy functioning ecosystem. And one of the reasons for this is that they bioaccumulate mercury within their tissue, meaning that it's methyl mercury that is from the atmosphere, settles into the sediment of lakes, and it's fed up through the ecosystem through benthic insects into fish, and then loons eat the fish. And so because loons are eating fish, their mercury levels tend to be higher than other organisms inside lakes. And even now, some people in some states are using loons actually in their mercury load to indicate whether or not fish are edible for humans. And here in New England, our loons actually have the highest amount of blood mercury of loons across the country. And so when I say loons here in the Northeast and loons of New England, what I'm talking about is this population that's genetically isolated and was actually taken out of, it used to go all the way down into Pennsylvania and New Jersey, some reports, but then in the late 1800s, due to hunting and egg destroying, the population for breeding loons was pushed into Adirondacks, New Hampshire, and Maine, until about the 1970s, where something really cool happened on its own. A pair of loons made their way down, they think, from New Hampshire into the Quabbin Reservoir. And from there, just slowly started expanding across Massachusetts and then re-entering into Vermont. And now we have a healthy population of loons in Maine, New Hampshire, New York, Vermont. Massachusetts now has approximately 40 breeding pairs, and Connecticut had their first breeding pair, I think, would be 4 years ago now. So loons on these aquatic ecosystems experience a lot of different stressors. So they're predated on. So loon eggs are a healthy snack for raccoons, apparently, because they love getting into the nests. Human disturbance is an issue, especially recreational human disturbance. A lot of these loons like to nest on ponds that we like to go wakeboarding on, water skiing, and boating activity. And unfortunately, this can disturb loon nests. Contaminants are an issue. Like I said in the beginning, that they all have high mercury load. They also deal with lead contamination from swallowing sinkers. They get into conflicts with each other. And so loons are really interesting that they're very territorial when they're reproducing. And it's estimated, I think, about 60 acres of lake for each pair of loons. And if it's less than 60 acres, this is where loons will get into conflict with each other. And we've done some loon necropsies here in the lab, and it's very interesting. When you open them up and look at their sternum, almost every adult loon has sternum punches that we think are from other loon beaks hitting their chest from when they're fighting. Water fluctuations are an issue for common loons. A lot of these loons like to nest on reservoirs, and so we control the water of reservoirs, and so the fluctuation can flood nests. Climate change as a whole, that also it causes water fluctuations with increased rainfall. And then finally, disease, which is something that we look at in our lab here. And common loons are actually impacted by a lot of different diseases. So Aspergillus, which is a fungal pathogen, it's usually like a byproduct of some other stressor. Loons don't like to beach themselves, but when they're really ill, they'll beach themselves, and that's where we think they pick up this fungal pathogen from breathing it in. Botulism outbreaks happen sometimes. Lead poisoning's a big issue for loons. We find in our necropsies that a decent amount of them are dying of lead poisoning still. Emaciation, that one actually is sometimes caused by mercury itself. So no loon's ever been dead from mercury poisoning, but mercury does have some neurological effects and can make it difficult for them to feed themselves. Ectoparasites, nematodes, which are like intestinal worms, and then finally the disease malaria, which is what I'm going to be talking about tonight. And so if you didn't know this, loons do die of malaria. So my advisor, Dr. Ellen Martinson, along with Dr. Inga Seigord at the pathology lab in New Hampshire, found a female dead loon on Umbagog up in New Hampshire that had died of signs of malaria. So malaria, the disease, is actually caused by a parasite, which I'll be talking about in a little bit, called Plasmodium, that's vectored by mosquito diseases. And these malaria parasites are not new. So malaria itself can impact a lot of naive hosts. So these are hosts that have never encountered the disease before. And so it's still going on across the world. So the native Hawaiian honeycreepers in Hawaii have been dealing with it for the past couple of decades. And recently, as of 2018, the Kiwis in Auckland have been dying of the disease malaria. And so what is malaria? So malaria is a disease that's caused by parasites. And avian malaria is a group of 3 parasites that are usually lumped together called Hemoproteus, leukocytozoan, and Plasmodium. And all 3 parasites are blood protozoans that are transmitted to birds through different types of insects. So Hemoproteus, which is on the left there, is transmitted by biting midges. Leukocytozoan is transmitted by black flies, and then Plasmodium is transmitted by mosquitoes. They are found in every bird on every continent, or they're found in most bird species on every continent except for Antarctica. So they're pretty common parasites. And they're only considered pathogenic in those naive hosts, like I showed before. And a question I do get a lot is these avian malaria parasites are not harmful for humans. However, Plasmodium is a parasite that can infect humans. We do have our own primate Plasmodium, but it's different than the one that infects birds. And moving forward for the rest of the talk, I'm going to be talking about Plasmodium, the parasite, and Leukocytozoan, the parasite, because these are the 2 avian malaria parasites that common loons have. We have yet to find common loon with the Hemoproteus parasite. So the Plasmodium parasite's life cycle. So these parasites have a really complicated life cycle. So they start off inside the insect. So for Plasmodium, it is a mosquito insect. And a lot of people don't know this, but mosquitoes are actually, majority of their lives are spent in aquatic environments. And so when they come out as adults, that's when the females need a blood meal to reproduce. And so to take a blood meal from a bird, that's where the first time she takes a blood meal where she can get infected with Plasmodium from another bird species. And so the insect gets Plasmodium from a bird and then re-injects that parasite into another bird host where it goes into a tissue. It reproduces inside that tissue. And then finally, when the parasite knows that it's time to be sucked up by an insect, it enters the red bloodstream, goes inside a red blood cell, and that's when the effects of the disease malaria can be felt. Leukocytozoan has a similar parasite life history. It just looks a little different, and its vector is black flies. And so this is New England, so we're all very familiar with black flies. They come out in the early springtime. These little insects also spend most of their life in water, but they rely on ephemeral streams. And what's really fascinating is that loons have a really interesting biological history with black flies. And so if you've ever seen a breeding female loon or male loon, when they're sitting on nests, their heads are usually covered in black flies. And what I have here on the bottom is a really cool study that came out in 2006 by a master's student, where she took different decoys of different bird species and set them out and found that there's a specific black fly that likes the pattern of loons. So it targets loons specifically, which I think is really interesting. And because it has a black fly that targets it, it's not shocking then that loons have their own leukocytozoan parasite. And so this parasite is specific to loons. And down here on the bottom, I have blood smears. So that means we take a slide of blood and we put it under a microscope. And what you're looking at is these bigger cells with a big circle in them. Those are red blood cells because birds have nucleated red blood cells. And the other purple, dark purple ring around that other cell, that is the parasite leukocytozoan, which is really cool. So they enter the red blood cells, they change the shape of the red blood cells, and they feed on the cell until it's time to get sucked up by an insect and complete its life cycle. And what's fascinating is not only do common loons have this leukocytozoan parasite, red-throated loons have been found recently to have the same exact leukocytozoan parasite, which makes us think that maybe they co-evolved with these parasites because they have a black fly, who's the vector of the parasite, who targets loons specifically. And when they target the loons, they inject this parasite, and this parasite specializes on loons themselves. And we don't know if loons have lifelong infections of these parasites or if they're able to clear these parasites, which is still a question. And so in the beginning, I talked about how loons have a history with mercury burden, and it's actually known that every common loon that we've sampled has a circulating mercury burden that can be detected. And so the question we have is there's literature out there that's in mice showing that mercury exposure, specifically in mercury exposure, has a response to Plasmodium infection. So mice with Plasmodium, the higher their mercury levels, the higher the amount of Plasmodium parasites circulating through them. So we call that parasitemia. And the reason we think this, or the authors thought this, was that mercury impairs host resistance to malaria infection, and that mercury-exposed mice, because of this, had higher parasitemia. So it's really cool that in other vertebrates, there's an interaction between heavy metals and these parasites. So now I'm going to talk about the data set that I have. And a big thank you again to these common loon working groups who have amassed these huge data collections of blood samples over the past 30 years. So in my particular data set, I have 731 samples. And the map you're looking at is every single dot is an individual loon blood sample that was taken over those years. Darker spots means more areas from those loons. In my data set, the samples were collected from 2006 to 2023. All the birds were adults who were caught during the breeding season. And this is because adult loons are extremely difficult to catch. The only time you can catch a healthy adult loon is when it has chicks or it's on nest. And because they're such good parents, you kind of use the chicks to lure the adults, and then you scoop them up in a net. And every bird has their blood taken, so I know the location that the bird was caught, I know the date they were caught, and then we know the mercury within that blood sample, mercury levels. So here's the questions that I asked. The first question is, what is the prevalence of these parasites? So I wanted to know, where were the positive individuals? How many of them were positive? Did they have co-infection? And was there different years of positives? The second question I have is, does environmental mercury exposure affect host susceptibility to parasite infections? And that's again due to other studies that found that mercury burden interacts with parasite infection. And the methods that I used. And so blood was collected either in the field, so I got to go out a couple times and capture loons with these crews. And then I also use their huge amount of archived blood samples that they have in their freezers. And what I do is I screen them through a multiplex PCR, which is a molecular technique where you can target the DNA of the parasite within the blood sample and then run a gel out, which is what the picture is here. And it shows you if you get a band, it means you're positive for the parasite. If you don't get a band, you're not positive. And then what's really cool is these groups had the foresight to take blood smears. So when they took a blood smear, it means that I can go in and actually see how infected the birds are. So now let's get into some results. Oops. So we'll start with the first one. Where are the infected loons? So here on the left is the loons that came up positive for Plasmodium. So this is combining all the years of the data set. And so of the 751 birds, 86 of them have come up positive for Plasmodium parasite. And you can see it's decently spread out. So we have the Rangeley region, that's the area of Maine close to Canada. The center of New Hampshire, we call that the Lakes region, so that's Lake Winnipesaukee. In Squam Lake area, a couple positives have come up in the Adirondacks. A couple positives have come up in Massachusetts in the Quabbin Reservoir area. Now for leukocytozoan. 271 birds came up positive for leukocytozoan. And similar regions with a lot of infections happening, majority in the Rangeley region, the Adirondacks region, the Lakes region, and then the Belgrades, which is right up there near Augusta. So that's where they're infected. Now, can loons be co-infected? That's not something we really thought of before, but the answer is yes. And so of those infected loons with Plasmodium, 48% of them also were infected with leukocytozoan, which is really fascinating. And we don't really know why. And then this picture that I just put up is a blood smear of a bird that actually had a co-infection. And so the circle in the blue is showing the leukocytozoan parasite, and the circle in red is the Plasmodium parasite inside the red blood cell. So it's really cool. And I'm also noticing that under the microscope, birds that have Plasmodium infections, their leukocytozoan infections are extremely high and the parasites clump together. And we don't really know why that's happening as well. So the next part is the prevalence of the birds that are infected. So what I'm showing here is a graph where on the y-axis we have the prevalence, so the amount of individuals that came up positive for that year. The x-axis is the year collected. I have it separated out by state, so each color is a different state. And then I have it separated out again with the top being leukocytozoan parasite and the bottom being Plasmodium parasite. So you can see that Plasmodium has a lower prevalence than leukocytozoan. What I think is really interesting that is if you look at the trend or how the prevalence goes, there's a difference per year. And the reason for this, we're not sure, but we think it might have something to do with the lifestyle of the vector. So mosquitoes and black flies, you might have noticed, have differences per year with the abundance of the amount of the amount of mosquitoes. So this year we had a really high mosquito year, but we didn't seem to have the same high black fly year. And so maybe this is why loons also potentially have different infections per year. I'm also starting to notice a trend that leukocytozoan seems to be increasing in certain years. And so if we account for the fact that there's uncertainty with probability of infection or the prevalence of infection each year, years with infections that are considered high are having higher infections than other years, which is really interesting. There's also something called spatial autocorrelation with these parasitic infections. So in disease ecology, there's something known as spatial autocorrelation, which basically is how close an individual is to another infected individual and whether or not that determines their infection. So this is how we get hotspots, so areas where a lot of individuals are infected. And we don't see this for Plasmodium, and that's most likely because Plasmodium, only 86 individuals were infected, and it's less than 10% per year in high years of infections. But in leukocytozoan, we're seeing a different trend. So this is the year 2019, and you can see that 53% of the birds sampled that year were positive, and those positives were all in similar areas, which is really cool. And so the Lake region of New Hampshire and Rangeley region of Maine, the majority of individuals in those regions came up positive for leukocytozoan infection. And what's even cooler is if we take a step closer to the data, it's actually breeding adults on the same pond coming up positive for the same infection. And so it could be potentially that these adults are passing the infection to each other. And so that's question one. So we're going to move on to question two, which is, does environmental mercury exposure affect host susceptibility to parasitic infections? And to do this, I did some modeling. And so the factors that I was interested in was the year the bird was caught, the state they were caught in, the lake they were caught on, and then the interaction between sex and mercury. And the reason that sex and mercury is an interaction is because it's known that males have higher mercury burden than females. And that is due to the fact that males eat more fish and they eat larger fish, and therefore they accumulate mercury at a higher rate than females do. And so the model results. So for Plasmodium, the best fit model, it took into account year, state, and the interaction between sex and mercury, and not lake location. That didn't matter for the model. And that found that mercury exposure does not significantly affect the probability of infections when it comes to Plasmodium. Now for leukocytozoan, it's a different story. And so the model best fit had year, the state the bird was caught on, the lake the bird was caught on, and then the interaction between mercury and sex, and found a significant relationship between mercury exposure and the influence it had on the infection risk of leukocytozoan, which is really interesting. And so to look at that a step further, so this is a logistic regression. On the y-axis, it's the predicted probability of infections. And on the x-axis, it's blood mercury burden, separated out by female in blue and male in green. And you can see on the graph that males with lower mercury have a higher probability of having an infection than males with high mercury burden, which is fascinating because originally we thought higher mercury means lower immune system, means increased likelihood for infection. But that's actually not what we're seeing. We're seeing the complete opposite, which is males with really high mercury burden having less of a chance of having an infection. So what is going on? Well, we've developed a hypothesis. Well, first, this is a box plot where on the y-axis we have blood and mercury burden in parts per million, and the x-axis we have the infection status separated out by males and females with no infection and males and females with infection on the right, just visually showing it. And what I'm going to do here is add in this line. And what this line is, it's 3.0 parts per million circulating mercury. We have papers showing this is what above that causes behavioral changes in common loons. So loons are considered to have a high mercury burden if they have higher than 3.0 parts per million. And you can see in this box plot, it's showing that females rarely have a mercury burden above 3.0, where males can reach that and still reproduce. So what do we think is going on? Well, if females have above 3.0 parts per million, they most likely won't reproduce, which means they wouldn't be in our data set because we wouldn't be able to catch them. They wouldn't be a reproducing female. However, we do have data showing that when males have high mercury burden, they will not nest switch with the females as often as they would if they didn't have high mercury burden. So loons are really fascinating because when they nest, they actually switch off with each other. They share responsibilities in nest duties, male and females, and they switch about every 6 hours. And there's some really cool studies where people have timed the nest switching and found that birds with higher mercury burden don't switch every couple hours. They'll take longer or they won't switch at all, which means the female sits on nest longer, therefore potentially getting bit by more black flies. So in summary, common loons can be infected with both Plasmodium and leukocytozoan. Infections have been detected in Maine, Massachusetts, New Hampshire, and New York. Leukocytozoan infections are more prevalent than Plasmodium infections. And for both parasites, infections vary by year. So some years have high infections, some years have almost no infections or very low infections. We can identify hotspots for infection for leukocytozoan, but not Plasmodium. So Plasmodium appears to be appearing randomly in this population of loons. So we can't guess what year it's going to be. We can't even guess the location. It just pops up. And finally, males with higher mercury levels are less likely to have leukocytozoan infections. So the next steps, so I have a lot of next steps to take. So the first is I want to screen more samples. So the Biodiversity Research Institute has samples going back to 1994. And so with that, I want to ask the question whether Plasmodium has always been present in the New England population, and then it's just recently we're finding them dead from the disease malaria, or has it always been there and there's some other reason why they're dying of malaria now, or we're finding them dying? I want to incorporate more climate variables into my model. And so because I'm seeing the variation between different prevalence of infections between different years, I really want to look at rainfall, temperature, and other factors that can impact the vectors and see if that is potentially what's driving these yearly differences. And finally, I have a really cool data set of loon species from across the world. So I have some Pacific loons, I have some Arctic loons, and I really want to see if these loon species share the same parasites as the red-throated loons and the common loons. So that is all I have, but I would love to take as many questions as you guys have for me, whether it's on the talk or loons in general. I know you guys have Eric coming in a couple months, who is a loon whiz and expert. But yeah, thank you.
Great, Erica. Thank you so much. And your final slide is lovely. We love dogs and loons.
Oh yeah, he used to go on the boat with me for my loon surveys.
So we have a couple questions here. If the study is of New England, I'm wondering why it did not include any samples from Vermont, where loons are also known to nest. Just curious.
That is a really great question, and the reason for that is Vermont sampling stopped around 2005, and it's just because Vermont, the loon biologist Eric, who you'll have speak, does an amazing job. He works super hard, but catching loons is extremely time and resource intensive. And so unfortunately, they don't ban loons here anymore. But we did start 2 years ago, so maybe we can start getting samples.
Do you suspect that the rate of infection with either parasite is increasing as the climate changes?
That's a really good question. Plasmodium, because the amount of infected individuals is so low, that kind of question is hard to answer, but I do think leukocytozoan seems to be increasing. And so in years that we are finding positives, the amount of positives we're finding is more, which is interesting.
Is there a tolerable parasite load in loons which will not result in mortality?
Yeah, that's a really good point. So these parasites, when they co-evolve with their hosts, so when the hosts are supposed to have them, they're considered non-pathogenic. And so when I look at blood smears of loons with leukocytozoan infections, they're at really low levels, except when they have a plasmodium infection, that's when I'm finding leukocytozoan parasite increases. And so maybe there's an interaction there, and it could be that their immune system didn't evolve to handle plasmodium infections, but it did for leukocytozoan. But that's a good point. We've never found a loon dead from leukocytozoan, and loons have come up positive for plasmodium that we know are still alive today.
Okay, another question. If you've seen any studies on the prevalence of malaria across population loons outside of New England, and whether it is more populous here compared to elsewhere in loon ranges.
Yeah, that's a really good question. So our lab is the only one looking at this parasite in loons. We did have a master's student who graduated 2 years ago, Alyssa Newhouse, who did screen loons from the Minnesota area and found that they are infected with these parasites, but at much lower levels than New England. And so I do have those data sets, and I would love to screen them myself as well.
One question I had was, and people, please, while I'm talking, feel free to populate the chat with some more questions for Erica. You mentioned that the males with lower, let me see if I get this right. The males with higher mercury loads, because they weren't sitting on the nest as long, were having lower leukocytozoan loads, correct?
Yeah, that's the hypothesis I'm putting out there
Okay.
because it doesn't make sense to me. Like you would think
Okay.
that if their mercury burden was high, then their immune system wouldn't be functioning at its best, and so therefore they would have a more likely chance of being infected with these parasites.
So are
But
we seeing I guess my question is, are we seeing that the females for the, because it sounds like you were capturing birds and you could tell you probably have, you know that you have a sample from the same nests as those males. Are you seeing a correspondingly higher load in the females of the nests with those males?
that is my next step, yeah, because that would be really cool to see. So I do know pairs because people target pairs, and yeah, I just haven't looked at the data, but that's a really good point. If I can match those two together, and that's the only explanation we have so far. It must be potentially with the nesting behavior or the parental behavior, because when you see loons with black fly infections, it's usually the birds sitting on nests because they can't dive and get those insects off of them. So they just take the bites while they sit on nest.
So I know you shared one observation of a bird, of a loon dying from avian malaria. Are there other examples across New England of it killing loons, or is it primarily making loons just sick?
Yeah, so when they first discovered it, they thought it could potentially be an epidemic, like it is in island systems. So, like the Hawaiian honey creepers and the Kiwis. It's not that bad for loons. I mean, they are dying, but it's a couple individuals every year. So, not enough to impact the population. My advisor, Ellen Martinson, does do pathology work with other researchers. So she does report deaths in other species of malaria infections. And so it could just be that it's occurring and we don't know it, because the only time you can diagnose the disease malaria is if you have a freshly dead bird. So loons could be dying of malaria, but then by the time they get to the necropsy table, they're too decaying for us to tell.
Why do you mention males eat more fish than females? Is there a reason for that? And are male and female birds generally very different in weight?
Yeah, they are different in weight. Females are lighter than males. So when I say eat more birds, it's mostly weight. Bigger birds eat more, and they can also eat larger fish too. And so these loons on ponds love yellow perch, which vary in sizes. But yeah, that's a good question.
Is there any research being done on prophylactic measures to malaria in loons similar to those employed with humans?
Yeah, good question. There is not, and it wouldn't be possible until we can get a grasp on where these infections are occurring and at what rate. Because it truly is showing up in the data set randomly. I can't predict the ponds that it's coming to, which is interesting. Um,
So would you say, based on your work, both in the lab and your prior work with loons that the main sort of, if we as people who care about loons sort of want to support loon conservation, like what are the primary things we, not necessarily your average Joe, but like people in general should be doing for loons? Yeah, what are the main threats, I guess?
I think the biggest threat that people can help advocate for is not using lead sinkers. That is the reason some of the adults die every year. Because that's a bias to my data too. And a lot of the loon work, we focus on breeding individuals. It's because these are the individuals we see. They're on the aquatic ponds. They're also the ones reproducing. And yeah, if we're losing some of these adults every year because they swallow lead, they don't do well once they have lead in their system. So I think that's the biggest thing we can advocate for. That and boat recreation. Boat recreation, we did a necropsy recently where it was a chick who was hit by a boat, and so that still occurs. They're still waking on nests, and so I think just speaking on your lakes when you see it happen.
Let's see. You mentioned some possible re-expansion of loon ranges. Is that still transpiring in spite of these parasitic threats? And I guess I would add, in spite of sort of climate projections of where we see, it seems counterintuitive of loons going south, expanding south when we sort of know that climate is a threat to them writ large. So, yeah.
Yeah, yeah, that's a good question. I know the populations are considered stable, but at the loon meetings I go to every year, chick production is down. And so we are seeing adults returning to their ponds, but we're not seeing chicks coming out of those breeding years. And loons are really difficult to study because they're really long lived. I think the oldest breeding loon is close to 40 years old now. So we don't know the ages of them. But I did work on the translocation program in Massachusetts where they're trying to expand their range further south because they used to be south. Um, so yeah, they are slowly making their way there. I mean, Connecticut just had their first breeding pair a couple years ago, which is really cool. So I think it's a matter of time before they reach Rhode Island on their own.
So they're expanding, but not necessarily, we could see range retraction if these chicks, aren't coming back.
Yeah, yeah. And I don't know if we'd even see the results of that until a couple decades from now, just because there are a lot of adults still circulating around.
Okay, another question. Have you looked at whether parasite infections are more common in birds that also have other conditions like, oh, I'm going to butcher this. You said it at the beginning.
Aspergillus?
Yes.
Yeah, yeah, that's a really good question. Unfortunately, so when we do necropsies, when we open a bird up, we see a lot of aspergillus. And the thing about aspergillus, it's usually a weakened immune system is how the fungus can kind of take control of the bird. So you can't diagnose it until the bird's deceased. I mean, they do throat swabs sometime, but there's aspergillus on everything. So it's really hard for us in the lab to determine whether the bird had aspergillus or you infected it on accident. So yeah, that's a good question. We don't know. Mercury is the body condition proxy we're using. Other than that, it's hard in loons because you can't use weight. In other bird species, they use body condition index, so they look at the relationship between weight and different morphological features. The loons are really interesting because some of them will stop flying during the breeding season, and so they lose like muscular atrophy, and so they actually lose weight until they go back. So yeah, that's a good point.
Let's see. What is the typical lifespan of loons and how many generations might they be able to raise? Obviously, it sounds like that 40-year-old is at the extreme end of things, but what's sort of typical?
Yeah, that's a good question that I don't know if anybody's gotten a solid answer to, or at least I don't have a solid answer to. So in the data set I have from BRI, we have individuals who reproduce almost every year, some who show up in the data set every 10 years. And so it's kind of chaotic. The loon's life cycle is really interesting. So they reproduce on aquatic ponds, but they're actually seabirds. They spend the majority of their lives on the ocean. And so some individuals will come in at the age of 5 to aquatic ponds to start trying to reproduce, and some of them won't even start trying to reproduce until they're 12. And so it's unclear. We do know, though, that it's important to these ponds for the adults that come back year after year. those are the ones producing chicks. Because loon chicks are hard for the adults to raise. Not only do they only have 2 eggs and that they raise them over 12 weeks, but also loons are extremely territorial and aggressive. And so adult loons kill each other's chicks. And so it takes a more experienced adult to raise a chick to adulthood.
Taking this new information into account, do you have any recommendations for updates to PPE when handling? I assume that means when handling loons.
I'm sorry, what's the
Well, if our question asker Lauren maybe wants to provide a little clarification, I think the question is, does any of this knowledge sort of change how you as biologists handle and work with loons in terms of
Oh,
protecting yourselves or the loons, etc.?
yeah, so the parasites can only be spread through the insect vector. So we can't spread the parasite. But PPE is always brought up in conversations because of HPAI. And so loon researchers are always looking at that and following those guidelines. Thankfully, it doesn't seem like HPAI outbreaks have occurred in loons. If they do get avian influenza, it's usually the low pathogenic one, and they're getting it on ocean. So yeah, it doesn't
For
change.
people who don't know, HPAI is high pathogenic avian influenza.
Oh yeah.
Yeah. Okay. Great. You mentioned the data set only included breeding adults, but I'm really curious if the parasites have an impact on chicks and younger loons that is different than in the adults, or whether they get infected at all.
Yeah, that's a really good question. So I do have chicks in my data set. I just didn't include them in any of my modeling. Chicks do get infected with leukocytozoan, and it tends to be chicks on ponds where the adults are infected. I have not had a chick come up positive for a Plasmodium and Parasite, which is interesting. And the reason we think that is because we think they're getting their Plasmodium parasites also sitting on nest. And so these are birds on nest getting bit by mosquitoes.
Okay. I'm gonna wait another second to see if we have any last minute questions. People do the teacher thing and just awkward silence it for a second.
Yeah, I will say our lab has a website. My advisor, Dr. Ellen Martinson, is head of the Loon Mortality Project, which is taking in dead loons and looking for their malaria parasites, which is a little bit different than what I'm doing. And so feel free to check our website out, and I can put it in the chat if you want to keep updated on all the critters we're screening for parasites.
I did get one last question in. So you had talked about chicks getting infected. Wouldn't the chicks also be getting bit by the mosquitoes when they're in the nest?
Yeah, that's a really good question. So loons are fascinating that when the chicks hatch, the chicks immediately stumble into the water. And so if we think potentially that the mosquitoes don't like being in the center of the lake the same way the black flies are, or it could be that mosquitoes come out before the chicks hatch. So loons sit on nests depends on where they are between April and June. So it could be that late nesters, their chicks aren't getting bit by mosquitoes the same way. But yeah, that's a good point. We don't know. Yeah. But I have seen loons hatch, and yeah, the chicks kind of just fall into the lake and just start swimming.
All right, well, so we'll stay tuned for more results from you in the future. People can subscribe to our newsletter. Maybe we'll share out something from you in the future. With that, I just want to thank everyone for attending tonight, and to Erica for her wonderful presentation and her work on one of our most iconic species here in Vermont. Please stay tuned on our YouTube channel for a recording of this talk, as well as our other past talks. And check out the links that I dropped in the chat for our upcoming online and in-person programs. And with that, I would like to say good night to everyone and thank you so much.
Thank you.